Good morning, everyone. I'm Vinny Lokhande. I'm a senior research associate at Growdea Technologies, and I'm truly delighted to welcome all participants in this webinar. It will always privilege to engage with professional researchers and students who are passionate about the life science innovation. Before begin the webinar, I briefly introduce who we are. So, Growdea Technologies is founded by Dr. Avinash Mishra at IIT in 2018. And as we evolved, we moved our base to INESCOM Innovation Center, Gurgaon. So Growdea are specialized in offering computational biology solution, which is tailored for both academia and industries, supporting early ages discovery through technologies like molecular modeling, structure-based designing, and AI-driven predictions. One of the our most milestone is our in silico based platform analog, which is designed to accelerate the streamline the discovery of novel therapeutics and analog therapeutics. Analog brings together the data science, automation, and biological insight in the integral solution. To share more about this vision and how we are shaping the future of drug discovery, I am honored to hand over this session to the founder of Growdea Technologies, Dr. Avinash Mishra. Avinash, you can continue.
Yeah, okay. Thank you, Vinny, and thank you to everyone for coming to this webinar series. So before I start, I would just have a very small announcement to make, that this would be like a series of webinar. This is not just a one webinar that we are conducting and then we will vanish off. So it is more like the series. Where the multiple webinars we are going to arrange after every fortnight. So it's like after 2 weeks, we will have the webinar. And let's also understand that what is the core objective of the webinar. And of course, all the webinar would be free for everyone. So I would, like insist all of you to join all the future webinar too. And the core objective of the webinar is definitely to introduce that what we are actually doing right now. Introduce our company, introduce our product. But more than that, the underlying objective is that how the computational biology can help experimentalists in drug discovery area. Okay, and we will also try to understand the limitation of it. When we are talking about the advantages that we have in the computational biology, we should also understand that what are the limitations. Otherwise, the story is incomplete. Okay, so I'll just share my screen, and then I will. So in today's webinar, so we will meet again, as I said, every fortnight we are going to meet. So today we are going to talk about the product, which just Vinny talked or just briefly talked about analog. And now I'm going to take you into slight detail, not like the full detail of it, but into some detail of the analog product. So let me just share my screen. So I hope that all of you can view the screen now. And of course, you can use the chat box to like if you have any question in between. And what I would also like advise or suggest that Vinny, can you just share your email ID? Or maybe you already have received her email ID when you received the meeting link. So if you have any question further, you can drop the message to Vinny, and then we will look upon the concern or the question that you have. And being very honest, because we are, running a commercial entity, we may not be able to go very deep into each and every question, but we will try to answer some of them. Okay. Because of course we have a limited time and limited resources, so we may not be able to entertain all the questions, but of course we will go for a few questions that we can answer in a short time. Okay, so this is the drug discovery platform Analog that I'm going to talk about. Before that, Vinny, I think already told you that this is a company called Growdea, and in Growdea we are working on complete in silico, that is the computational biology method in life science. And of course, the umbrella is so big, but we are just covering small molecule protein design, genomic data analysis, or secondary analysis. So we started with a service offering company, and we are still offering the solutions, but now we also come up with a product which is analog, and this is something that we all are here to talk about. The problem statement that we are addressing at this point of time is the drug discovery, of course. And in drug discovery, we all know that it is a highly challenging area, and one of the indication that if somebody asks you that why you think that and how you think that drug discovery is challenging, then if you see a lot of people in any domain are interested and into it, then one of the indication could be that the domain itself is not that much challenging, right? Especially when you have to spend your money, spend your effort. Okay, so if it is a high risk kind of domain, like the drug discovery, then it is clearly you can see its projection in the market, that you will find very less commercial entity will go for it, because there is a high risk, and especially in Indian context, because we brought up in such a way that we always hide ourselves from taking risk, right? So this is one of the high risk area where 80 to 90% of the chance that a people or a entity or a company or an organization can fail in the clinical trial. Okay, uh, while it takes almost like 10 to 15 years of time. And 10 to 15 years of time is a huge time, right? Like, decade, more than decades. And then 2.8 billion of dollars. I'm not talking about INR. So it is dollars that you need to spend. So, I mean, if such kind of parameters are there, years, money, risk, why anyone would go easily for it, until unless they have a very clear vision, passion, and appetite to take risk. Then but one of the way that we can do is by mitigating the risk. I'm not saying that we can eliminate the risk, we can like solve the problem, but we can always mitigate the risk. We can always cut down the risk. Okay, and that is what the computational biology can offer to you. So always, I don't know how many of you are from the computational background, or how many of you are from the experimental background, but I would say that don't take computational biology as the solution which will solve everything, what is lying in front of you. But also don't underestimate the power of the computational biology. And this is the correct way of taking things. That don't think that this is the solution, but also don't think that this can't do anything. Okay, so computational biology is also the same. It can solve a lot of things. It can give you a solution for multiple items, but it is not the solution for every item. Okay, so you can cut down the risk, cut down the time, cut down the effort, cut down the cost involved, but you may not be able to, uh, give the final solution using the computational biology. But of course, this area is evolving, and it has been evolved at a very high rate in the last 5 years, and especially because of the AI and machine learning. And um, before that, the things were addressed using the physics-based principle, like docking, computational folding of a protein, or a structure prediction of a molecule, or molecular dynamics. All these are the physics-based or chemistry-based. Now, AI has come into this domain, and AI is a data-based domain, where it defines its own rule based on the data. Okay, so whatever the data you feed to your model, then it will define the rules for that data set. So this is interesting, because when we had physics-based principle, the time when I was in my PhD, of course, machine learning was there, but not like the scale it is right now. So at that time, we were talking more about a chemistry-based principle, a physics-based principle, and apply it on the molecule. Then what happens is that you define the principle ahead, and then you apply it on the molecule. Here in AI, it is not like that. It first take the data, understand, and then make the rule. Okay, so that's the difference, and both has limitation, both has advantage. So that's the thing, and that's why we decided that we will not go just for one. We'll go for both of them, because both of them are required. Okay, so it is like, uh, if we combine both of them, physics based and AI, and machine learning, then you will see the complete story. And this is what the analog can bring or can offer to you. So this whole platform, analog, is a graphical platform. It's a very easy to use platform. You can, and of course, this is the first webinar. So I may not go in the live, but I will definitely show you the live product. So everything is very easy to use, because I understand that many of us may not be that efficient in programming, which is okay, because that is not the field from where we came. So many of you could be a very good chemist, or some of you could be a very good life science experimentalist, or biotechnologist, or biochemist, right? Or microbiologist. But maybe the programming is not your cup of tea, which is fine, because you have something which is your strength. But nowadays, as I shown you till now, that computational things are very much like required in your science practice. So how you will do it? Because your background, your training is in, let's say, microbiology. Now somebody asks you that you need to do some computational work to get it done in a quick time, and a less with a less resources. So their analog can help you out. Okay, so you can use analog and do your stuff quickly, efficiently, and with a decent accuracy. Now, analog, as I said, has 10 different modules. So these are the 8. Um, there are two more below this. And now we also have another one. So now the slide is, like a bit older. So there are now 11 modules, and we are keep on working. So what the objective, as a company, that that how we can develop each and everything, at least in the drug discovery, which can be used by the experimentalist in a very easy way without going deep into the uh, programming or command or syntax. Okay. And take the example of ChatGPT. So what ChatGPT is doing for you is it is making your life easier, right? So you need not to understand that how it is giving you the answer, right? And you never bothered about it. You just bothered about it that is it giving the answer, and then if it is giving, is it giving the correct answer or not? Okay. And slowly and gradually, the ChatGPT is also, getting trained more and more on the real data set. And then the accuracy of it has been improved. Like 1 year back when I and this is 100% real. 1 year back, when the ChatGPT, I think ChatGPT introduced in maybe 2 years or 2.5 years back. Uh, and in the initial ChatGPT, when I put this question, what is 13 plus 12, it gave me wrong answer. Right? Can you imagine this? The ChatGPT on which we rely a lot nowadays, and there is nothing wrong in that. Um, but like 2.5 years, it was not like the way it is in the current scenario. And after 2.5 years, it will be much stronger than way it is right now. So because it is getting trained, and same way, we are also getting evolved, and we are also putting more and more modules with to our analog, so that people can use this in silico drug discovery almost like the same way they are using the ChatGPT. Okay, this might be like a very far analogy for comparing ChatGPT with NLP, but just give me that liberty to take it. But this is how the idea has been evolved. Okay, now the product is in the market, and of course it's a licensed product. Okay, so in the webinar, I will also talk about some scientific things that are actually necessary to know, like what is QSAR, what is decoy, what is drug, what is binding score. So I will also give you some training on the computational drug discovery, and at the same time I will also tell you that how you can do all these things on analog. Okay. So there will be a science part and there will be an application part. So as I said that there are 10 different modules with it, and I will go in today for all the modules very briefly. It is not like that I'm going very deep into every module, and I will take every module. So suppose I have 11 modules, so at least 11 webinar is sure, right? And maybe more than 11 if one module can take more than two webinar. Okay, so stick with it. And by the end of all the series of the webinar, I'm sure that you will get a very good understanding that what the in silico drug discovery is. And if somebody would be interested, they can reach us for like the purchasing this application. Okay. Of course, I mean, I won't be hesitate to say that this is also a promotion of our product, right? Because we want it to go to everyone, those who are using it, using drug discovery. So we want to promote it, and this is one of the way. But at the same time, I'm not just being centered towards my product, but I'm also like diversified towards the scientific domain of it. Okay, so the first part is QSAR, where we are saying that how we can relate, and of course the detail will be in the later webinar. But QSAR saying that how you can relate the structure of any compound with its activity. Okay, and this tool can allow you to build that relation. Okay. And now the word Q is standing for quantitative. Okay. And S for structure, A for activity, and R for relation. So quantitative is structure activity relation. Now, structure activity relationship is quite clear. You know that. Okay, I have to relate the structure with the activity and build a relationship. Now, what kind of relationship it would be? Okay, um, and I'm not talking about the human relationship, which is so complex. I'm talking about something which can be interpreted. Okay, and when we are saying about the relation that can be interpreted, then the best example is the mathematical relation, right? So when you have something like a equation, when you say x plus y, x plus 3x, or x plus x square is equal to 34. It is easy to understand because these are the numbers, and believe me, there is nothing easier than numbers to understand. Okay. Other than numbers, everything is difficult to understand because everything then subjective. Then everything other than the numbers will have a different perspective for different people, right? But when I say 34, it has the same meaning when you say 34, right? So your 34 will be same as my 34, but your good will be different from my good, right? So that's why, and that this is called, and this is the reason why the word called quantitative in this, because it's a mathematical relationship. So we allow user to build a mathematical relationship between a structure and the activity. We take simple smiles from, and if you guys know, and I would also request, you know, people who are very new to In-Silico, that try reading something right in between, because now we are going to meet after 2 weeks. So in that period, you can read that what is smiles, what is PDB, what is or what is STF, all this. Now, but for time being, I'm telling you that the smiles is like a simple signature of a molecule, right? Like a string, the simplest form to represent a molecule, and it takes that and then it calculates almost like 1,500 different chemical features. These are 1D features, 2D features, 3D features, and I can explain you, uh, that what are those features means. Okay, maybe slowly, gradually. Okay, and and then those features are being related with the activity and to build a mathematical model. That is how the mathematical model is built, and why it is built, so that the next time if there is some, so building is only on the known data set where the activity is known and the structure is known. Now, in the unknown scenario, when the structure is known, but the activity is not known because we have not done the experiment, then the mathematical model that we built can predict that activity and help you to avoid the experiment, or like assist you to do the experiment. Okay, so that is why we need QSAR. Now, the other module is drug molecule generative, which is for generating new compounds. Now we have generative AI, and this is one of the generative AI module. So the generative AI, and one of the common example is nowadays that you see images that are AI generated, even the images of humans, which is AI generated, right? So if the images of the humans can be generated by AI, uh, then then the molecules can also be generated by AI, and this is on the same principle that it generates the molecules. Now, in this module, we have a pre-trained model. What is pre-trained model? Is the model which is already been developed by us using our own data set. I mean, own data set doesn't mean that that is a proprietary to us. Own data set means some data set that we collected from whatever the source is. And we collected the ChemBL data. It's in million. And then we trained it, and then we built a model. So we have a pre-trained model, but at the same time, we also give this option to users to build their own model. So all the modules that we have is not just restricting user to use our model, but we are also allowing users to build their own model. So that is how we have done it. Then we have drug decoy identification, where this is like um, that something. What is decoy? I think you guys know it. It's an English word. Something which looks like the actual thing, but it is actually not, right? So in drug discovery also, we have decoys which may look like a drug, but it is not. So then we used machine learning, once again, a graph neural network, and built a drug decoy classification machine learning model. But because the decoy and a drug is very specific um, to the protein, right? So like for one protein, one molecule could be a drug, but the same molecule could be a decoy for some other protein. So it is protein is specific. We know that, right? Because it is protein specific, it means the machine learning model should also be protein specific. We can't say that this is the universal model for drug decoy, because then it is like all the proteins are same, but which is not the scenario. So it means that you need to have a different machine learning model for different proteins. And we have almost, like, if I'm not mistaking, almost like 113 or 115 different proteins model. And these are the proteins which are commonly used as a drug target. So we have a pre-trained model, 115 model for 115 protein. But suppose your protein is not in the list, then you need to develop your own model. Okay. Um, and then test it. The next module is the protein drug binding, and what exactly it is that when we are performing docking, many times, believe me, many times, because docking is a mathematical algorithm, right? And protein binding, protein drug binding is not a mathematical phenomenon, right? So it is, but we can't do anything, because mathematics is a tool by which we solve a lot of problems, and protein drug is also one of it. Um, but sometimes you will find that the docking program can bind anything with your protein, but the moment you go to the experiment, it is not working. So we built a ML model to bring that experimental information and predict the IC50 for you. Okay. But this is only when you dock the compound and give the docked compound to our model. Once again, we have a pre-trained case, and also allow user to build their own model. Okay. Then we have protein drug interaction. So this is also giving you the IC50 or binding kind of estimate. But this is from the sequence. The earlier one was from the protein and drug docked structure. And this is just simply from the sequence itself. So you just need to give the sequence of a protein and a smiles of a compound, and it will take it and it will predict or estimate the IC50. Now, if you are thinking that how accurate it would be, so compared to the structure based, it will be definitely less accurate. But compared to the structure based, it will be, like high in speed, or it is very rapid. So so like we have some test, like in diagnosis, you definitely have heard it, like antigen antibody test. um, I'm not right now referring to this. I'm just taking one example. Like in COVID, right? In the COVID scenario, we had some PCR test and we had some, I think, antigen test. And antigen test was rapid test, right? While the other one, PCR, was taking some time. So when you have to do something rapidly, you have to compromise with certain level of accuracy. It is uh, it is there everywhere, wherever you are doing any job. Certain level of accuracy you need to compromise, but this is highly rapid. It can predict the IC50 in a minute for thousand of compounds. So but of course it will have a lesser accuracy. So what you can do is you can just use it for the screening, and you have million of compound, you can just screen thousand of them from using this tool. Okay, so this is and then you can go for docking, and then many other things that we have. This module, antigen antibody, which is the next one, is based on the protein-protein interaction. This once again takes only the only the sequences of the proteins from the antigen and antibody, and predict the KD for it. It also allows you to change your protein, or to design or make some modification in your protein, and then go for binding again. So it, in some sense, and on all these process are automated. So in some sense, it is saying that you can design a novel antigen, for example, which can trigger the antibody production, right? Or which can bind with the antibody. Okay. So and a vice versa also. Like you can design or develop some new antibody, or by modifying the existing one. So this is for that. We have actually, there is frag ana and doc ana, but the order should be reversed. I'll change it next time. So let's first talk about the doc ana. Doc ana is a docking. So you just have to give your molecule in the PDB format, and we allow for single docking, we allow for multiple docking. In multiple docking, you just need to have the smiles and the protein PDB structure. In single docking, you can have the PDB file format for compounds and for protein. You need not to worry about the binding pocket. It will automatically detect the best binding pocket and docket. You can ask the program to generate 10 conformers, 20 conformers, whatever the numbers, and then it will generate it, and then it will give you the docking docked structure. Then you can also generate the 2D images for protein drug interactions, like you might have seen in a lot of publications. Then you can download the docked complex also to open it in any viewer, right? After docking, we have fragana, which is like once the molecule is docked, if you want to improve the docking or the binding by changing certain part of the molecule, the fragana module will allow you to select some part of the protein, some part of the molecule, and click it by, which means that you're selecting it, and then it will go back to the fragment library or a functional group library, and then attach different functional group at that particular point automatically, and then bring the top 20 to you. So it will, so in nutshell, that it will modify the existing ligand, and evolve it as a new ligand with a better binding affinity. Okay, and then you can download the new it, and then you can redock using docana and see that is it really true or bad or whatever it is. Okay. Then finally, we have a GROMACS-based molecular dynamics simulation program where all different kind of simulation. We started with protein ligand, protein peptide, only protein in water solvent. And now we also have umbrella sampling. We also working on replica exchange, which will be very soon the part of the product. So all complex kind of simulation. Now you will ask that, what is new here? So new is that all different kind of simulation you can perform in a very easy way. Believe me, and we are going to do it at some point of time. You need not to be trained on this. So my whole objective, or in fact the objective of complete team, is that you should not put a lot of time getting trained on using the tool. You know, like I believe that if you have to be trained, or you have to be like spend lot of time on training, then there is something wrong with the utility of the tool. I mean, it should be easy to use. I mean, you also love to start using any machine or any application from the next day. Who cares about the manual, right? So if such kind of tools are made, it will be easier um, for the non-computational, and even for the computational biology people. Of course, they would be much more comfortable. So this is like you can run, start from the protein ligand, prepare it at different pH. You can prepare, you can go for different protonation, then you can add water, put it in a box, minimize it, equilibration, NVT and NPT, go for the production at the end, and then finish it. And then we have a trajectory analyzer, which will allow you to analyze the whole trajectory that is being generated in the molecular dynamics. And this analysis could be done in a different matrix like RMSD, RMSF. Uh, yeah, so I'll come to the question later. Okay, I'll come to the question later. Maybe I'll keep 10 minutes. I'll just try to finish it in next 10 minutes, and last 10 minutes I'll keep it for the question. If the questions are not answered, as I said, you can just drop email. Okay, but when you are dropping the email, don't forget to put your full detail of yourself at the bottom. Like where you are, what you are doing. I'll be happy to know that one. So okay, fine. And you can analyze, you can build a free energy landscape, or principal component analysis, or binding free energy. All these calculation, you can do it in a very easy and effective way. Okay, and finally you will have a report. You can download the report, you can download the images, you can download the tables, everything. Okay. Okay, so this is what we have. We have one more thing, and I'm going to talk about briefly on that part also. So direct users for these tools are scientists, R&D professionals, PhD candidates, and even the master candidates. Okay, I'm not sure about the bachelors right now, but the way the standards are being uplifted, maybe at very soon we would say that yes, the bachelors also need such kind of tool. This is another thing which is the external part of the analog, which is not directly coming with the analog, but also the extended arm of analog, which is called octatox. And this is the toxicity based module where you can predict the toxicity, and these are the human toxicity based prediction. Okay, so it is not the animal toxicity based prediction, but the human toxicity based prediction. So we are having the neurotoxicity, cardio, hepato, nephro, and pulmonary toxicity, which can be predicted. Okay, it is explainable toxicity. You can also generate a new compound which might not be toxic, like this. So this compound is toxic, but and this is the part which is the toxic element of this compound. You can ask the software to generate a new compound. It will generate a new compound which will not be toxic. Okay, so that is also a part of it. So as I said, the data which we used in the training is the human toxicity based data, not the animal toxicity based data. So it is much more relevant. So finally, what we deliver something like this. Okay, so this is the hardware, and what we thought is that if we are just delivering the software, it might not be equally acceptable, or highly acceptable, sorry, highly acceptable. Why? Because all these modules that I talked about needs a very hard GPU based machine, and somebody may say that I don't have any GPU machine, and then how I will use your software. So there are two ways. One is if I bring it to the cloud, where I'm using my own resources, and you submit your job and get the result. And the other way is this. So I started with this way. I thought that I will provide the complete workstation, something like this, which is in picture, with a minimum like 32 GB RAM, Ryzen 5 processor, NVIDIA RTX 3060 GPU, and 1 TB storage, many other things, but these are the um, most important component of the hardware. And then analog is pre-installed. So you can just use it from the next day when you have it in front of you. And we are also not stopping you to uh, use it for some other purpose. So suppose you are working on NGS, you can use it. It's a 12 core processor, high in RAM, so you can use it for NGS, and you can use it for some other purpose also. This is the team that we have. Some of us are here in this meeting. We have some competitive advantages because the tool is not just built, like uh, in a month's time. We took almost like 4 years effectively, because we first wanted to know that whatever the algorithm we are building, whatever the pipeline we are building, is it really working in the experiment? And we started providing solutions to different clients. They started testing our compounds in the experiment, and they saw result in the experiment for our molecules. So that helped us to gain confidence and to build this platform. Okay, now the same thing which we are doing, anyone else can also do the same using analog. Okay, we also definitely have price edge, because it's a uh, indigenous product, so definitely it would be less pricey than any other foreign source product. Then it's a data security edge, because we are giving the whole workstation, so you can do whatever you want on this workstation. We have several successful stories in the past. Some of the stories still we are doing. These are the case studies, and all these case studies are not just the theoretical work. Everything is complemented with the experimental, whether it is a small molecule design, or a peptide design, or a small molecule design for anti-cancer, or a vaccine development. Everywhere the things are complemented with the experiment. This is for the toxicity. Okay, so we have our website, and this is the first talk with all of you. We have our website, we have our LinkedIn page. So I would ask Vinny to send all these details, like our website, our publications, our tool cited, our LinkedIn page, everything to all the participants, and I will request all of you to go and visit and join us, and if you have any question, you can always reach to us. uh, Being a commercial entity, we have to do the business. There is no if and but on that side. But this commercial entity is also originated from the science domain. So that's why we always have a explanations or the understanding for the science. Okay. Now, before stopping, what I will do is I'll just stop this sharing, and because I'm not going to today demonstrate or demonstrate each and every module, but I will demonstrate. I'll just show it that how it looks like. So just give me a second, and I'll share the another screen, which is for the tool. Okay, so this is the tool that I was talking about. Okay, analog. Uh, Once you have the machine, of course, I said that we will give you the machine and the pre-installed analog, but that doesn't mean that if you say that I have already the machine, I have the GPU machine, and I just need your platform, we can also do that. So no issue on that side. Then uh, you will get all these. So you can see that there are 11 modules. Umbrella sampling is also here, which was not a part of the my presentation. Then we have QSAR, antigen, antibody, and you just need to go to any module. So suppose I go to SimAna, which is simulation ANA. So here you just have all the options or all the steps at the top, and then every step will have options here. Okay, so you just need to click. Okay, so suppose I need to remove water, I just need to say remove water and submit it. Okay, you have a download sample file, so how your file should look like, you can see it here. How to use is the manual, so you can read the manual if you want that how to use the tool. Okay, and then go to the next step every time and perform your simulation, solvation, ionization, minimization, equilibrium using NVT ensemble or NPT ensemble and productions. Okay. You do perform, and then there is a protonation, as I said, that what type of protonation you need. And then once you perform the simulation, once the simulation is done, you will have all the files on your machine or on your system. Then you go to trajectory. Here you can calculate like what is the RMSD, RMSF, hydrogen bonds, SASA, radius of gyration. So every time it will ask you that, okay, what is the group one for binding energy? Because binding energy is between two groups. Which group you want to, and once you upload the simulation file, you will also see the groups information. It will tell you that, okay, this is this group, this is this group, this is protein, this is ligand, and you just have to give the numbers here, like 12, 30, and go for calculating the binding energy. And then you can say that, okay, I need a plot. Right now there is no plot, so that's why we don't see it. But once you run it, you will see the plot here, and you can download the plot. You can have this scatter plot for PCA or FEL. You can have the 3D view plot. Okay, different type of plot, whatever is right now being accepted in manuscript. Okay, so and similarly, you can go to any tool and then you can use it. We are going to do some exercise slowly, gradually as we move ahead on this. Okay. Now, what I will do is I'll just now check. The docking in the analog is offering right now. It's a rigid docking, but we have like simulation, so you can perform the simulation for getting it more accurate docked pose. Okay. Uh, Now what I would request all of you to maybe we can start building some group. I don't know. I'm not sure that can we make a Google group or some kind of group. Any VM type access. Uh, what do you mean by VM? It's like a virtual machine, or what actually you mean by that? Virtual machine. Okay. Yeah. So we give a trial period, but that is for the case where the people is interested or they want to buy it at some point of time. Uh, that kind of thing is possible. And uh, if you want to use it for a day, maybe you can. If you are in NCR, you can also come to our office. You can use it. Okay. And if you have a plan, or if your department has a plan, you can actually go for it. We can assign one machine to you. And so for analog, of course, it's a commercial product. So you need to buy it for using. You can have some free trial, but at the end you need to buy it. But uh, the other part of this whole webinar is the scientific understanding uh, of the concept, like docking, simulation, QSAR, umbrella sampling, all these scientific things. That is also be a equal part of this. Okay, and then we will explain that how you can do the same thing using analog. Okay, so I will try to make some group. I don't know what's not WhatsApp, I think, or I don't know if you suggest WhatsApp. We can do the WhatsApp group also. But for WhatsApp group, we need the numbers. I don't know if we have your numbers or not. But if we have numbers, we can make a WhatsApp group, because WhatsApp is bit faster way of communication. So we can do that. Okay. And if you have any question or any concern or anything you want to, be in touch, once the WhatsApp group is there, you can just post there also. Yeah, but sometimes you write something on a WhatsApp group, and then other people post something else, and then your message goes away. So that's the problem of that. So you can still reach to us. Binny's email ID she already shared, and you already got when you receive this invitation for the webinar. Okay. So I will just stop here, and then we can meet again. And that's why I'm saying that follow us on LinkedIn, and and then you will get to know that when the next webinar would be, and we are going to start with QSR. Okay. Okay. So thank you. Thank you, everyone. We can stop this now, this webinar, and we will meet again. Okay. Thank you. You can leave the meeting now.